Hi everyone, welcome to lesson 2.7, DNA structure and history. This is our last lesson of unit 2, so make sure to fill out your guided notes and vocabulary as we go through this lesson. First up, we have a video on DNA, so go ahead and pause here, watch the video back in Blend, and answer these two questions. Now that you've watched the video, let's go ahead and move on. So nucleic acids are composed of nucleotides. We learned that back in lesson 2.3 throughout our biomolecules unit. We know that DNA and RNA are examples of nucleic acids, and we know that nucleic acids store and transmit genetic or hereditary information. But this question, how do genes work, this is going to come up more and more, especially as we get closer to unit 4, genetics. But we have to understand what genes are made of in order for us to understand that question, how do genes work? So today we're going to examine the discovery of DNA and then the structure of DNA. In 1928, Frederick Griffith was trying to figure out how bacteria make people sick. More specifically, Griffith wanted to learn how certain types of bacteria produce the serious lung illness called pneumonia. So in his experiment, Griffith injected mice with 4 different samples of bacteria. When injected separately, neither the heat-killed, disease-causing bacteria nor the live, harmless bacteria killed the mice. The 2 strains injected together, however, caused fatal pneumonia. From this experiment, Griffith inferred that the genetic information could be transferred from one bacterial strain to another. So today we know that this transforming principle that Griffith observed was the DNA of the S strain bacteria. While the bacteria had been killed, the DNA had survived the heating process and was taken up by the R strain bacteria, which is how we get that transformation principle. So the race to discover the cause of transformation was on. And next up we have Oswald Avery's experiments in 1944. So Avery and his team isolated various molecules such as proteins, lipids, carbohydrates, and RNA from the heat-killed bacteria and treated them with destructive enzymes. But the transformation still occurred. When DNA was destroyed, the transformation did not occur. So by observing that bacterial transformation, Avery and the other scientists discovered that the nucleic acid DNA was the source that stored and transmitted genetic information from one generation of bacteria to the next. So next up we have bacteriophages or bacteriophages, whichever way you like to say it. So a bacteriophage is a type of virus that infects bacteria. When a bacteriophage enters a bacterium, it attaches to the surface of the bacterial cell and injects its genetic information into it. The viral genes act to produce many new bacteriophages and they gradually destroy the bacterium. When the cell splits open, hundreds of new viruses burst out. So why is it important that we know what a bacteriophage is? Because of Hershey-Chase. So Hershey-Chase experiments were in 1952. They studied a bacteriophage made out of DNA and a protein coat. They wanted to determine which part of the virus, either the protein coat or the DNA course, entered the bacterial cell. Their results would either support or disprove Avery's findings that genes were made of DNA. So they grew bacteriophages with radioactive sulfur and phosphorus isotopes that could act as what we call markers. So the results said that nearly all the radioactivity in the bacteria was from phosphorus, the marker which was found in the DNA. So Hershey and Chase's experiment with bacteriophages confirmed Avery's results, convincing many scientists that DNA was the genetic material found in genes, not just in viruses and bacteria, but in all living cells. So at this point, we know the roles of DNA. Okay, we know it has 3 major roles: storing information, copying information, and transmitting information. So we're going to get into kind of the specifics of storing, copying, and transmitting whenever we get into Unit 4. But for now, we're trying to still understand the structure of DNA. So the components of DNA. So scientists knew DNA was the genetic material, but they needed to figure out how it was stored, copied, and transmitted. DNA is a nucleic acid made up of nucleotides joined into a chain by covalent bonds. Okay, so the phosphate group of one nucleotide covalently bonds to the 3' carbon of the sugar of the next nucleotide in a repeating pattern called the sugar-phosphate backbone. And nucleotides have 3 parts, which we have seen before: a phosphate group, a sugar, and a nitrogenous base. So if this seems confusing to you, like what does this aspect mean here? Okay, so it tells us here is a phosphate, here's the sugar, which is connected to the next nucleotide, phosphate, sugar, next nucleotide, phosphate, sugar. This here is what we call the sugar phosphate backbone, okay? And this is the piece that is covalently bonded, okay? So far we know the structure of a nucleotide and their three parts, and how one strand of DNA, okay, has covalent bonds between nucleotides, and that is what makes up the sugar phosphate backbone. Now we have, in 1949, Erwin Chargaff discovered that the percentage of A and T are almost equal in a DNA sample, and similarly the percentage of G and C were almost equal. The observation that A is equal to T and G is equal to C became known as Chargaff's rule, or the base pairing rule. So base pairing is the principle that states that the bonds in DNA can form only between adenine and thymine and between guanine and cytosine. So here's our first kind of check for understanding. If a species has 35% adenine in its DNA, what is the percentage of the other three bases? So even though the answer is up on the screen right now, kind of pause and think about how this was calculated. So since we had 35% adenine, we automatically know there has to be 35% thymine because these two are going to equal. Okay, once you add those two up, you get 70, okay, and then subtract that number from 100%, which you get 30, and then you split that into between guanine and cytosine, the other two bases, and you get 15% of each. So here's a check for understanding. Go ahead, pause here, and try this on your own. Okay, so the first one is really similar to that last question that we just went over. A DNA sample contains 23% cytosine. What are the percentages of the other nitrogenous bases? So, because of base pairing, we know if there's 23% cytosine, there has to be 23% guanine. So far, that is 46%, these two added together. Now, subtract that 46% from 100. Okay, then you get 54% left for adenine and thymine. And because these two numbers are equal, you divide that 54% by 2, and you get 27% thymine, 27% adenine. Now, one way you can check yourself is to add up all four of these together. Okay, 23, 23, 27, 27. If that equals 100%, okay, that is good. And remember, C has to be equal to G, T has to be equal to A. Number 2, a DNA strand has the sequence here. Okay, write out the complementary strands. So this is something that we haven't done yet, so I put a hint in that last question. But this is going to be something that you do later on. So we're going to go ahead and practice now. Since we know A is equal to T, okay, whenever it says to write out the complementary strand, you're literally just matching the base pairing rule. So we have A to T, T goes to A, C goes to G, G goes to C, and so on and so forth. Okay, so if you see that phrase, write out the complementary strand, it just wants you to use base pairing to match the next DNA strand like this. So far we know that DNA from both humans and bacteria obeyed Chargaff's rule, but nobody really understood why or the importance of this base pairing. Okay, so then in comes Rosalind Franklin. So in the early 1950s, Rosalind Franklin began to study DNA using X-ray diffraction. So first she purified a large amount of DNA, then she stretched the DNA fibers in a thin glass tube so that they were parallel. She aimed a powerful X-ray beam at the concentrated DNA samples and recorded the scattering pattern of the X-rays on film. By itself, Franklin's X-ray patterns did not reveal the structure of DNA, but it did carry some very important clues. So here is her picture. So this X pattern here shows that the strands in DNA are twisted around each other like coils of a spring, or a shape that we call a helix. And the angle of the X suggested that there were two strands in the structure. So at the same time Franklin was doing her research, Watson and Crick were also trying to understand the structure of DNA. In 1953, when they saw a copy of Franklin's X-ray patterns, they were able to build a model that explained the specific structure and properties of DNA. The pair published their results in 1953, when Franklin's paper describing her X-ray work was also published. Watson and Crick's model of DNA was a double helix, in which two strands of nucleotide sequences were wound around each other. So here's more on this double helix model. This model explains Chargaff's rule and how the two strands of DNA are held together. So first is this property that we call anti-parallel, or DNA runs in anti-parallel strands. So the two strands of DNA run in opposite directions, allowing the nitrogenous bases to contact in the middle. The nitrogenous bases are held together in between with hydrogen bonds, okay, which are slightly weaker than covalent bonds. And it's very important that the strands are held together with hydrogen bonds because they must be able to be separated in order to replicate. And then this model, this double helix model, also explained base pairing rules, or Chargaff's rule. So here is a diagram. Okay, so here we have our nucleotides. Okay, in the middle, our nitrogenous bases are bound together with these dotted lines, and these dotted lines are representing the hydrogen bonds. These red arrows are showing that anti-parallel property. So this strand, it shows running up, while this strand's running down, right? So they're still parallel to each other, but anti just means running in opposite directions. So here's a check for understanding. Go ahead, pause here, and try this one on your own. So this check for understanding wanted you to kind of practice writing out the bonds, getting the structure, and the structure of your nucleotides. Okay, so first we have had this question quite a bit, but here's a nucleotide. You have a phosphate group, a sugar, and a nitrogenous base. Okay, the second part is showing the sugar phosphate backbone, right? So sugar phosphate backbone, or those covalent bonds here between the sugar of one group and the phosphate group of a next. In the middle, our nitrogenous bases are joined with hydrogen bonds, these dotted lines here. Okay, and then if you want to see this kind of the double helix model, here you have your 4 bases. You see your base pairs in the middle bonded with hydrogen bonds, and then your sugar phosphate backbone, this on the outside. And then you see this 3' 5'. So this is something that you're going to look at later, but 3' and 5' are basically just a way of labeling the ends of the DNA strands. So you notice that the 3' end of one matches with the 5' end of the other. And the most important thing that you understand right now from this is that is that anti-parallel property, right? If one strand's running in one direction, the other one's running in the other direction. So now we have reached our exit ticket. Go ahead, pause here, and try out these questions on your own. So first, the 3 roles of DNA were storing, copying, and transmitting information. Number 2, draw a nucleotide and label the 3 parts. So here is that nucleotide for reference. Number 3, A pairs with T and G pairs with C. Number 4, the nucleotides were joined by the hydrogen bonds in the middle. Number 5, the sugar phosphate backbones made up by covalent bonds. Number 6, Rosalind Franklin used X-ray diffraction and discovered that DNA has 2 strands. And then number 7, using Franklin's X-ray diffraction images, Watson and Crick were able to create the double helix model. So that is the end of lesson 2.7. Make sure to check your notes, get your 3 vocab terms down from this lesson, and thank you for watching.